Glad to be here exposure my job about transposable elements in Neotropical species of Drosophila called Drosophila amaguana. Most of the eukaryotic species has the variation in the genome size, and this variation always is associated with a large amount of DNA. Usually, this amount of DNA is associated with repetitive DNA, such as transposable elements, simple repeats, even satellite DNA. The insects have often been used to study transposable elements because these sequences have an impact and a key role in their evolution. Inside the arthropodal genomes, all the species that belong to this classification have a high variable T content because, for example, here we have a Belgica Antarctica that show a genome size of 99 megabases that show a lower T content compared to, for example, Locusta migratoria that has the highest genome size in all the insect species, showing a percentage of TE content nearly to 65%. Here in this table shows many Drosophila species and the respective repeat coverage or repeat content associated to transposable elements. It's important to know that in all the species, it's not possible always try to sequencing the complete genome. For example, in the last column, we can see there always exists a little variation about the genome assembled compared to the genome estimated through the flow cytometry. And here, for example, Drosophila melanogaster has been used widely to study transposable elements, showing that in this species exists a percentage around 50 to 20% of these repetitive elements, showing 146 families, including 165 sequences, using the Berkeley Drosophila Genome Project and the manual created T library databases. Also, in many populations of Drosophila melanogaster from Africa, these species have lower T content compared to other populations from other continents. So it's important to know that in all the species of Drosophila has the same content of TE in other parts of the world. For our study, we focus on Drosophila amaguana, that is a member of Mesopragmatica group. This group is an endemic group from South America. This species was discovered in the first or the beginning of the 21st century in a place that showed high vegetation, low luminosity, and high humidity. With this, we identified that the genome of Drosophila amaguana showed a highest value of size, 455.5 megabases, exceeding even the value reported until now of Drosophila sirtoloma, that is 400 megabases. That is the most higher reporting it in all Drosophila species. With this, we will try to identify the T content in Drosophila amaguana and try to understand what is the role of these Ts in the contribution of the genome size for Drosophila amaguana. The methodology that we used was assemble the genome with Mazurka, evaluate the quality or metrics of the genome assembled with a Python script called AssemblyStats, and then use Busco. And for the TE process, we use the novel program, the novel tools, EDTA, Repeat Modeler, and Resona TE to generate the novel TE libraries. With these de novo T libraries, we select only the results from EDTA and Repeat Modeler to do a manually curated library using the tool MC Helper, Manual Curator Helper 2, and try to analyze all the TEs created from this species. In the results, we can see in the first row the size of the assembled genome for Drosophila amaguana, that is 455.5 megabases, and the M50 value was around 21 kilobases. As mentioned before, in the second table we show three species that show different TE content associated to the genome size. In our results, with the novel tool libraries, using ETA, repeat modeler, and reactional TE, we obtain different results. For example, in ETA, we show a high value or a high percentage of LTR retrotransposons that was the most higher compared to the other tools. But for example, in repeat modeler, in repeat modeler, we can see that show the highest value of unclassified sequences. With this, we can see here in the plots different composition of retrotransposons or TE class 1 and DNA transposon or TE class 2. In EDTA, the percentage was similar. In the repeat modeler, we obtain more unclassified sequences, but in reactional TE, we show a low value of retrotransposons compared to DNA transposons. With the T libraries obtained from EDTA and repeat modeler, we do a manual creation of these libraries, obtaining 737 T consensus sequences, from which 716 was putative or putative new T families, because these families has never been described before, and only 21 TE sequences has been described previously in other Drosophila species. This shows a percentage or 39% of retrotransposons and 60% of DNA transposons. With this, T create library and incorporate into the libraries from Berkeley Drosophila project and the manual creator T library that was reported in a paper using two Drosophila species, which we annotate 972 TEs in all the assembled genome from Drosophila amaguana. With this, we can see that the genome of Drosophila amaguana contains a percentage of 46% of class 1 elements with 12 superfamilies and 53% of class 2 elements with 11 superfamilies. Here we can see in the bar plot the most abundant superfamilies of TEs annotated, or the number of copies of TEs found in the Drosophila amaguana genome. And we can see the highest bar corresponds to the elytrons, that shows a value of 30%, while, for example, signs and DIRS elements show the low copy number that even we cannot see in the bar plot. The bar is very high, very low. For example, other important results here, we have that tiers, LTRs, and lines and mites was other orders present in the Drosophila amaguana. In the tier orders, the superfamilies TC1 Mariner and Hat were represented with 19% of all the tier copies, while in the altier retrotransposons, Gypsy showed the half of all the copies from this order. While there were other superfamilies that show a lower number of copies, less than 200 or 100. Here we analyze the landscape of these superfamilies, and here we can see that between a divergence ranged from 15 to 25, exists an increased TE activity more than 4,000 copies. And this corresponds to order or TE orders like Elytron, TIRS, and LTR, suggesting that all these copies maybe correspond to other transposition events in this species. The highest peak in the activity we can see here, there is around 20%, and this divergence indicates that maybe at this point existed a transposition bars for elytrons. Analyzing the T insertion time in Drosophila amaguana, we also can see that most of the copies of the Ts, of all superfamilies Ts, was inserted approximately between 3 to 4.5 million years ago. But there are other superfamilies like TRIM, R1, R2, or PLAE that maybe appear some recently with the copies, low copies, less than 1 million. For example, here we can see some copies from PLE that show a short time insertion in this species. And also we have two other superfamilies that maybe was inserted in a range of years from 1 to 3 million years ago. With this, we can try to estimate the whole percentage of repetitive DNA in Drosophila amaguana. As we can see, using the manual curation library, we obtain a more real value of the TE content in the genome of this species. With this, we know or we estimate that the TE content corresponds to 21% of all the genome, that it's equals to 98 megabases, while the 66% consists of non-repetitive DNA. Here in the bar, we can see the blue color corresponds to the percentage of TE. The TE compositions was given by the most abundant amount of elytrons with one superfamily, the only superfamily now until now, and the composition was to conform by altiers with 5 superfamilies, tiers, and lines. We also currently we are doing some extra analysis to try to include in this estimated repetitive DNA content about the simple repeats, and even studying or try to estimate the value of the satellite DNA. For example, here the preliminary results that we obtained show that the simple repeats corresponds to 12% of the total genome, while the satellite DNA estimated from raw reads using Repeat Explorer was nearly to 5%. With this, we established some conclusions from this work, and the main is that the TA content was approximately 22% using an assembled genome of 455 megabases. And this TA content showed a high prevalence of elytron, tier, altier, and lines orders. And this is important because this is the first study in Ecuador and in other mesopragmatic species group that probably is an important point or important start point to allow more analysis and more research to understand what happened or what is the relationship between the TE content in the genome science. These results may offer a value insight for the future research into the role of all these mobile sequences that have in Drosophila amaguana and other species that inhabit in Ecuador and or in the Andean forest. But what is the next? Mainly, further detailed research and deeper analysis probably may improve significantly the estimated repetitive DNA content in Drosophila amaguana, and we are considering to expanding the study to other individuals or other populations, but this time using third-generation sequencing technologies to obtain long reads that maybe help us to detecting more complex sequences, may overcome some actual limitations because the genome was assembled using Illumina reads, and there are some papers that show a correlation or a relationship between the sequencing technology to the TA content using long reads, it's more possible to try to understand or try to identify more complete sequence of TE or more copies of TE compared using short reads. I would like to thank to all the institutions and the person that helped us to develop this work. And also I would like to thank to the Pontificia Universidad Católica del Ecuador by the funding support. Thank you.
Thank you, Manuel. We have time for just one question, very short question.
Well, I don't know if it's short, but we could talk afterwards. I don't know. I don't understand how do you make the manual creation step, because you first mentioned that you have like a consortium that before your work already has like manual created libraries of transposable elements, but afterwards you mentioned that you run different pipelines like in model, ETA, and you made another manual creation step. So did you use the previous manual created libraries, or you did new ones?
All the results were using the manual creation library. The first step of the methodology was using different de novo tools to identify the TA content by each tool. And with these TA de novo libraries, we did the manual creation. And with this manually created TA library, we annotate these consensus elements created from all in all the genome and use it to generate the plots that we analyze.